Well, we have to talk again microbial anatomy and physiology as we were talking about bacterial cell division, how one bacterial cell divides into two. For this, it is very important that every organelle must have to be divided into two equal parts. Uh, so for that, several genetic material, genetic mechanisms are involved in it. MREB is the one of the most important protein which is responsible to determine the cell bacterial cell morphology. So in prokaryotes contain a cell cytoskeleton that is dynamic and multifaceted. MREB, major shape determining factor in prokaryotes, forms simple cytoskeleton in bacteria and probably in archaea. So mRNA protein is shape determining either that bacteria will be cocci, that will be bacilli, that will be vibrio, or that will be spiral. Whatsoever shape of microorganism is going to be, for that it is responsible mRNA protein. What does this mRNA protein? It forms simple cytoskeleton in bacteria and in archaea. It forms spiral-shaped bands around the inside of the cell. So it must we have to be clear that mREB proteins are present or form spiral-shaped bands around the inside of the cell. So they are making a spiral band or a ring-like structure, a spiral ring. It forms a spiral-shaped band around the around the inside of the cell underneath the cytoplasmic membrane, not found in coccus-shaped bacteria. So mREB proteins are not present in cocci bacteria, but these are present in bacilli bacteria. These mREB proteins are present in bacterial cell in form of the spring inside of the cell membrane. So we must have to be clear that mREB proteins are responsible for the shape determination of bacterial cells. So these mREB proteins are present in bacillus type of bacterial cells and archaea, but not present in cocci round-shaped bacteria. And in bacillus-shaped bacteria, these are present in a form-like structure, and these are formed in a spiral-shaped band around the inside of the cell, underneath the cytoplasmic membrane. So mREB proteins, these localized synthesis of the new peptidoglycan and other cell wall components. So mREB proteins, where it is binds with cytoplasmic membrane, where it is adhered with the cytoplasmic membrane, they have function. They have to make new peptidoglycan in growing cell wall, as well as other wall components to specific location along the cylinder of a rod-shaped cell during growth. So here we can see this is FTSG protein, which will bind at center of the bacterial cell, and MRE protein, it is present in ring form, in a spiral form inside of the bacterial cell. So wherever it binds, where it sites, where it is attached with cytoplasmic membrane, uh, they will synthesis the cell wall over here. And in that result, what will be? The cell wall will be elongated, its size will be increased, its size will be bigger, and it will reach to its maximum level from where its septum will form and it will be divided into two bacterial cells. So mechanism we can see through here. And this is the electron micrograph of the images which has been taken by uh, Alex, that this is one bacterial cell in bacillus form. Due to MRAB proteins, it has been increased, it become large, and it has been divided into two. MREB and determination of cell morphology. Most archaeal genome contains FTSG and MREB-like proteins. Thus, cell morphology is similar to the seen in bacteria. So, archaea also have FTSG protein and MREB proteins, which are responsible for the bacterial cell determination as well as bacterial cell division. So how it is dividing? First, its size will increase. Its size will increase, and the size, how its size will be increased? Its size will be increased due to MREB proteins. MREB proteins at different sites they will synthesize new cell wall. They will synthesize new cell wall. They will increase. They will build new cell wall on existing cell wall by breaking. Peptidoglycan synthesis and cell division, production of new cell wall material is a major feature of the cell division. Production of new cell wall material is a major feature of cell division. In cocci, cell walls grow in opposite directions outward from the FTS0. In rod-shaped cells, growth occurs at several points along the length of this cell. So here I have showed you that at several points cell wall growth takes place during its enlargement. And in foci, it is over here just in cocci, cell wall grows in opposite directions outward from the FTS ring, as you can see here. This FTS ring and its cell wall is going to be increased from this side towards this side and towards this side. When it will reach at its maximum position, it will be break like this. This is electron micrograph. So, peptidoglycan synthesis and cell division, pre-existing peptidoglycan, we know that the cell wall is made up of peptidoglycan. Okay, how FTG and MREB proteins making new cell wall in existing cell wall. Pre-existing peptidoglycan needs to be served to allow newly synthesized peptidoglycan to form. So pre-existing peptidoglycan needs to be served to allow newly synthesized peptidoglycan to form. So pre-existing peptidoglycan will provide base for the formation of the new peptidoglycan. Beginning at the FTSG ring, small openings in the wall are created by autolysin. So before starting, how it starts? When mRNA protein is joined over there, it is binded with cytoplasmic membrane. So at beginning, the FTSG ring, a small opening in the wall are created by autolysin. A small opening will be, it will be cracked. It will be, and a small opening will be created in the inside of the cell wall. Or new cell wall material is added across the opening. First, autolysin will open a small space in bacterial cell wall. That open space will be covered, will be filled by uh, new formed material, new formed cell wall material, new formed peptidoglycan. Wall bands, junctions between new and old peptidoglycan, when molecules of the peptidoglycan will be attached in openings of the cell wall. So wall again will be formed and the junction will form between new and old peptidoglycans. here. So new cell wall synthesis takes place here, 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 here. So what they are doing, they are breaking a small fragment over here. They are opening a hole over here and inserting new molecules of the cell wall over here. So in detail, we can see this thing. Peptidoglycan synthesis and cell division. So how this takes place? So bactoprenol is the carrier molecule that plays major role in insertion of the peptidoglycan precursors. So this is working as a mason. Bact- Bactoprenol is responsible to carry peptidoglycan molecules from cytoplasm to the cell wall where opening has been formed, and it places it over there. So bactoprenol is 55 carbon containing alcoholic molecule bonds to it have ability bonds to N-acetyl glucosamine, N-acetyluramic acid, and pentapeptide peptidoglycan precursor. So we can see over here like this. Bactoprenol is a meson which is responsible to carry the peptidoglycan molecule N-acetyluramic acid and N-acetylglucosamine with phosphate molecules to that place which has been created but isolysine in cell wall. So it will keep these two blocks over there and its size will increase. So glycolases, enzymes that interact with bactoprenol, insert cell wall precursors into growing points of the cell wall, catalyze glycosidic bond formation. So this whole phenomena we have seen here. This is peptidoglycan. Okay, this is peptidoglycan. Growing point of the cell wall. This is peptidoglycan. Sorry uh, for interruption, please. We are going to carry on it again. Okay. This bactoprenol is responsible to carry the molecules from cytoplasm to bacterial cell wall site, synthesizing site of the bacterial cell wall. And this is the growing point of the bacterial cell wall. Here it has been created a space for the insertion of the new molecules, and new molecules of the cell wall has been carried out by bactoprenol to this place. Again, transglycosylase activity now from new coming molecules of cell wall and old molecules here will be uh, a bond that is called transglycosylation. Peptidoglycan synthesis and cell wall division, transpeptidation, final step in cell wall synthesis, forms the peptide cross links between muramic acid residues in adjacent glycan chain inhibited by the antibiotic penicillin. So at this stage, this synthesis, this transpeptidation can be stopped to stop the bacterial infection by various antibiotics. And this forms the peptide cross-link between muramic acid residues in the adjacent glycan chain inhibited by the antibiotic penicillin. So this thing, here we can see this transpeptidation between this and this molecule, it can be stopped. Uh, Population growth, the concept of the exponential growth, the mathematics of the exponential growth, and the microbial growth cycles. The concept of the exponential growth. Most bacteria have shorter generation time than eukaryotic microbes. Generation time is dependent on growth medium and incubation conditions. Sorry. So exponential growth, concept of the exponential growth is what? Exponential growth of the microbial population in which cell number double within the specific time interval. During exponential growth, the increase in cell number is initially slow but increases at a faster rate. The microbial growth cycle, batch culture, a closed system microbial culture of the fixed volume. Typical growth curve for population of cells grown in a closed system is characterized by four phases. Number one, lag phase, exponential phase, stationary phase, and death phase. So these phases we will study in detail in next coming class. Uh, Up to this, you have to take care, and you have to study these things in book, Prescott especially, or Plexar. You will find detail on. Have a nice time.